We are going to listen to Saranya Joshi she's from Garhwal University in India. So just to put you in the context, so she flew from India to here, I don't know, 40 hours or something, 35, 20, okay. And because of some visa problems, so she could arrive only yesterday. She was supposed to have a poster, but I think that we can wait a little bit for the coffee and listen to her work, given that her effort to be here. We really appreciate your effort for being here. So, okay.
Hello, am I audible? Okay. A very good morning to one and all present over here. I am Saranya Joshi from 𝑚𝑎𝑙𝑎𝑦𝑖 Gadwal University, India. I'm here to give an oral talk on the topic entitled as Comparative Analysis of the Complete Mitochondrial Genomes of two filarial worms, 𝑊𝑢𝑐ℎ𝑒𝑟𝑒𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎𝑚 with C. elegans. Now, uh, Wuchereria bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎𝑚, they are the parasitic worms and they belong to phylum Nematoda. And this work is basically concerned with the comparative genomics of their mitochondrial genomes along with the C. elegans. As they are causative agents of the disease elephantiasis, you can see the symptoms of the disease. This is the worst condition which arises um, uh, due to the effect of blocking of the lymph nodes. So coming on the comparative, this study, the comparative analysis of mitochondrial genome, which contains a minor but the important component of eukaryotic genome. Now, sorry. Okay, coming on the methodological part of this work, the very first step, that was the data mining. The genomic data of these three worms has been taken up from the NCBI database. And then for the identification of the conserved regions in these genomes, the MOVE software has been used up. After that, the identification of intergenic regions has been done with the use of Vista server. And in their complete mitochondrial genomes, the ORFs, that is the open reading frames, have been predicted using the ORF finder tool of NCBI. After that, the genomes were aligned by using the cluster-W program for finding out the sequential similarity using the multiple sequence alignment. For tracing their evolutionary history, Mega10 software has been used up. And at last, the 12 protein coding genes which are found in the mitochondrial genomes have been compared by using the cluster-W program. Now, okay. Now first the data mining step. This if we look at the nucleotide composition of these worms, they are quite similar in their ATGC content, and the size of the genomes are very similar to each other as well. Not depending upon the factor that the two worms are parasitic and the one is free living. Okay. Now this picture is basically showing the alignment results or the identification of conserved regions in the worm, which has been done by the MOVE software and using the alignment process. In the very first step, the Wuchereria bancrofti was aligned to C. elegans, followed by alignment of 𝑏𝑟𝑢𝑔𝑖𝑎𝑚 to C. elegans, and at last, Wuchereria and 𝑏𝑟𝑢𝑔𝑖𝑎𝑚 were aligned to each other to find out the conserved regions present in them. A total number of 13 conserved regions have been identified in this in these organisms, in which 7 regions were similar to each other, while 6 were unique in them. And in the identified conserved regions, those regions were further subjected to finding out the ORFs in them, so that we can find out whether these regions are coding or the non-coding part of the genome. So some ORS had the functions while others didn't. Now, for the identification of intergenic regions, a total number of 20 conserved intergenic regions have been identified, which are common in C. elegans as well as in Wuchereria bancrofti, and they showed the sequential identity ranging from 68 to 74%. And if we come to the intergenic regions, which are basically the non-coding part of the genome, so the smallest region was of 104 base pair, and the largest one was 333 base pair. In the alignment of 𝑏𝑟𝑢𝑔𝑖𝑎 to C. elegans, a total number of 22 intergenic regions have been identified, and the sequential identity ranging from 67 to 74. The sequential length 98 to 277 base pair. And at last, when 𝑘𝑒𝑟𝑒𝑟𝑖𝑎, 𝑏𝑟𝑢𝑔𝑖𝑎 and 𝑏𝑟𝑢𝑔𝑖𝑎 were aligned with each other to find out the intergenic, they showed a highest sequential identity of 87.5%. Now, this pictorial diagram is basically representing the total ORFs which have been identified in these species by using the NCBI ORF finder tool. And the very first step is of 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti, followed by 𝑏𝑟𝑢𝑔𝑖𝑎 and C. elegans. Now, among the identified ORFs, a total number of 63 ORFs were identified in 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti, 58 in 𝑏𝑟𝑢𝑔𝑖𝑎 and 40 in C. elegans, which were further subjected to protein blast so that we can find out their probable function, and about 32 in 𝑘𝑒𝑟𝑒𝑟𝑖𝑎, and these numbers are basically representing the functional ORFs through which we could assign some function by using the protein blast program of NCBI, and the remaining ORS didn't had any significant similarity through the protein blast tool. Now, if we compare the identified functional ORFs in these genomes, a total um, a total number of 17 ORFs are common in all the three organisms, and the remaining are unique in their species. Now, in terms of unique ORS, these are the list of ORS which are identified in 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti. Other than the 17 ORS, these are unique and have not been known to have in these organisms. This is the list of the ORS which have been predicted in 𝑏𝑟𝑢𝑔𝑖𝑎 melai, unique one, and the in the C. elegans as well. Now, coming to the multiple sequence alignment of these organism, cluster W program was used up, and we could find out that 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎 melai are more similar to each other since they have the alignment score of 88, while the aligning of C. elegans to 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎 melai sequentially gave a alignment score of 42, which is almost half of that. Now, coming on this diagram is basically representing the phylogenetic tree created by using the Mega 10 software, and the method used was maximum likelihood method. So, in this tree, the 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎 melai are closely related to each other, placing close enough, and the C. elegans is here, and but it could be find out that there is some sort of divergence from some point, because all the three organisms are actually coming from the same clade. Now, this table is basically representing the alignment score. A total number of 12 protein coding ORFs are 12 protein coding genes are found in the mitochondrial genomes of these worms. So we aligned these genes to each other to find out the sequential similarity in them. And it was find out that genes of 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎 melai are showing the highest alignment score in comparison to the other two alignment results. And at last, coming on the concluding remarks, no, 𝑘𝑒𝑟𝑒𝑟𝑖𝑎 bancrofti and 𝑏𝑟𝑢𝑔𝑖𝑎 melai are genetically similar to each other but are divergent from C. elegans, even though they have evolved from common ancestor. There was an early divergence of C. elegans, but still they contain the 12 same 12 protein coding genes in their mitochondrial genome. In the identification of conserved regions, we have identified some unique conserved regions in them, which may not code for some protein coding genes but are conserved at their sequential level. Some partial ORS have also been identified in these species, which have not been reported earlier, and the study identified these common conserved regions in the mitochondrial genome, which may explain their evolutionary and lifestyle differences. So these are the references, and I really want to acknowledge this the organizers of the meeting to giving me this opportunity so that I can present my work here and accommodating me in the last moment as well. I'm also thankful to Department of Biotechnology, Government of India, for providing me with the travel grant support so that I could come up here. And thank you so much. And this is our small lab team, which are working on different aspects of worms through in silico approaches. We are the dry lab work, and our supervisor, Dr. Sudhir Kumar, under which we are doing our work, and our lab partner. So this is Structural Bioinformatics Lab from India. Thank you so much. Namaste.
Okay, there is time for one short question. Okay, Luisa.
About tRNA genes, mitochondria are full of mitochondria-specific tRNAs.
Yes. Actually, this study is basically concerned with the protein coding genes, but there are 22 tRNAs which are found in their mitochondrial genomes. Maybe after that, we could work upon that. Thank you. Thank you.